Good morning, everyone. Welcome to today's presentation. I'm delighted to have you all here. Today, we will be discussing precision criteria in analytical method validation. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few minutes, we'll explore what precision means in analytical methods, why it's important, and how we can ensure our methods meet the necessary validation standards. So, let's get started and dive into this essential topic. Now, let's talk about why analytical method validation is so important. Essentially, it ensures that the method we use is reliable and accurate for its intended purpose. When we refer to USB 1225 validation parameters, we're talking about a set of key criteria that help us verify this. These include accuracy, which tells us how close our results are to the true value, and precision, which shows us how consistent our results are when repeated. Specificity is also critical. It means our method can distinguish the analyte from other components. Linearity confirms the method's ability to produce results proportional to the concentration across a range, which ties into the range itself, the span over which the method remains valid. Detection limit and quantitation limit define how small an amount we can reliably detect and measure. Robustness assesses how well the method performs under slight variations, and all of these factors together help ensure the method is suitable for its intended purpose. Ultimately, a validated method should be reliable and reproducible, giving us confidence in our analytical results. This is fundamental to maintaining quality and compliance in our work. Let's dive into the definition of precision according to USB. Precision refers to how closely a series of measurements agree with each other when we sample the same homogeneous sample multiple times under specific conditions. In simpler terms, it's about how consistent our repeated measurements are, how close they are to each other. Now, USB classifies precision into different types. First, we have repeatability, which is also called system precision or method precision. This is the variation we see when the same method is used multiple times under the same conditions. Then, there's intermediate precision, sometimes called ruggedness, which looks at variability when measurements are taken on different days, by different operators, or using different equipment, but still under the same overall conditions. Lastly, we have reproducibility, which assesses the variation when different labs or different analysts perform the measurements. Understanding these different types of precision helps us evaluate the reliability and consistency of our analytical methods across various scenarios. It's a fundamental aspect of ensuring the quality and robustness of our testing processes, and it ties directly into the overall theme of method validation and quality assurance in the lab. Let's talk about repeatability, or what we often call method precision. Essentially, this is about how consistently a method can produce the same results under the same conditions. It's a crucial aspect because if our method isn't repeatable, then our data could be unreliable. When we focus on repeatability, we're ensuring that small variations in measurements are minimized, which helps us trust our results. This ties directly into the overall theme of data quality and accuracy. After all, if we want our findings to be meaningful, they need to be consistent and dependable each time we run the method. So, emphasizing repeatability is about building confidence in the process and the results we obtain. Today, I want to walk you through an example of how we calculate precision in our method. As you can see on this slide, understanding precision is crucial because it tells us how many of the items we identified are actually correct. It's a key metric to evaluate the accuracy of our classification. In this example, we start by looking at the number of true positives. Those are the items our method correctly identified. Then, we see the total number of items our method labeled as positive, which includes both true positives and false positives. By dividing the true positives by the total predicted positives, we get the precision. This ratio gives us a clear picture of the reliability of our predictions. The higher the precision, the fewer false positives we have, which is especially important in applications where false alarms can be costly or problematic. Connecting this back to our overall theme, calculating precision helps us fine-tune our approach and improves the accuracy of our results. It's a fundamental step in validating the effectiveness of our method. Now let's move on to the acceptance criteria, specifically for the USB practice. When it comes to assay, we aim for a % RSD or relative standard deviation of 2% or less. This means our method should be very reproducible, ensuring consistent results across multiple runs. For impurities, the criteria are slightly more lenient, with a typical % RSD of up to 5%. However, it's important to note that this can vary depending on the level of impurities and specific requirements. Additionally, I want to emphasize that these criteria are not set in stone. They may differ based on our SOPs or regulatory guidelines. Always double-check the specific standards applicable to your particular analysis to ensure compliance. Ultimately, these acceptance criteria help us maintain the quality and reliability of our analytical methods, aligning with the overall goal of ensuring product safety and efficacy. Now, let's talk about system precision. This is evaluated by performing 6 replicate injections of our standard solution. The idea here is to see how consistent our system's response is when we run the same sample multiple times. For example, we look at the peak areas generated during each injection. If the system is precise, these peak areas should be very similar across all 6 runs. This consistency is crucial because it assures us that our measurements are reliable and reproducible. When we connect this to the overall quality of our analysis, good system precision means we can trust our data and make confident decisions based on it. Now, let's talk about intermediate precision. This is all about evaluating how consistent our method is under different conditions. We do this by intentionally changing several variables. For example, we might have different analysts performing the test, or we might run the analysis on different days. We also look at using different instruments or columns to see if those changes impact our results. To give you an example, imagine that on day 1, the mean assay result is 99.7%, and on day 2, it drops slightly to 99.4%. We then calculate the overall % relative standard deviation, or % RSD, by combining all the data from those different conditions. The key point here is that we want this % RSD to generally be less than or equal to 2%. That's our benchmark for acceptable intermediate precision, indicating that our method remains reliable even when variables change. This step helps ensure the robustness of our method across real-world testing scenarios. Let's talk about reproducibility. When we evaluate reproducibility, we typically do this during interlaboratory studies. This means that different laboratories are involved in the process. The idea is to see if the method produces consistent results across various settings. Reproducibility is especially important in collaborative studies, where multiple labs work together to validate a method. You'll find that reproducibility is usually a requirement when transferring a compendial method from one laboratory to another. Ensuring that the method remains reliable and consistent across different environments is crucial for maintaining quality and compliance. So, overall, reproducibility helps us build confidence that our methods are robust and can be reliably applied in different labs and situations. Now, let's talk about precision at different levels when using the impurity method. It's important to evaluate precision not just at a single point, but across multiple levels to get a comprehensive understanding. We start with the LOQ level, which is the limit of quantification, and it's where our measurements are just reliable enough to be considered accurate. Next, we look at the 100% level, which represents the target concentration we aim for in our analysis. Finally, we consider the 150% level, which is a higher concentration, helping us assess the method's robustness across a broader range. By evaluating precision at these different levels, we ensure that our impurity method is not only accurate at a specific point, but performs consistently across various conditions. This approach ultimately helps us ensure the reliability and reproducibility of our analytical results in real-world scenarios. So when we talk about methods, they can sometimes be precise but not accurate, or accurate but not precise. What does that really mean? Well, being precise means your measurements are consistent with each other, but if they're not close to the true value, then they're inaccurate. On the other hand, accurate measurements are close to the actual value, but if they vary a lot, then they're imprecise. This distinction is really important because it highlights that precision and accuracy are related, but not the same thing. Understanding this helps us improve our methods by aiming for both, being precise enough to get consistent results, and accurate enough to reflect the true value. In the context of our overall theme, mastering both precision and accuracy allows us to make reliable, trustworthy conclusions from our data. It's all about balancing these two aspects to ensure our results are not only consistent, but also correct. Let's talk about the importance of precision in our work. First, it ensures the consistency of results, which is crucial for reliable data. When our measurements are precise, we can trust that repeated tests will give us similar outcomes. This consistency is vital for making informed decisions. Next, precision helps us detect random errors, those unexpected fluctuations that can occur during testing. By maintaining high precision, we can identify and minimize these errors, improving overall accuracy. It's also important to recognize that precision is required for regulatory submission. Regulatory agencies demand precise data to ensure product safety and efficacy. Without it, our submissions might face delays or rejections. Furthermore, precision is critical for batch release in quality control. Only with accurate and consistent measurements can we confidently approve or reject a batch, ensuring only high-quality products reach the market. Finally, all these points tie back to our overarching goal, ensuring product reliability. Precision underpins the trustworthiness of our results, which ultimately supports the safety and effectiveness of our products. Let's wrap up with the importance of precision as a key validation parameter under USP 1225. Precision basically tells us how consistent our analytical results are when we repeat the measurements under the same conditions. We evaluate precision in several ways. As repeatability, which looks at consistency within a single run; intermediate precision, which considers variations within the same lab but at different times; and reproducibility, which checks how well the method performs across different labs. To quantify precision, we use statistical measures like the mean, standard deviation, and the percentage RSD, or relative standard deviation. These help us understand the variability and reliability of our data. Of course, all these calculations must meet predefined acceptance criteria to ensure the method is performing adequately. Ultimately, maintaining precision is crucial because it underpins the reliability and consistency of our analytical results, which is fundamental to quality control and regulatory compliance. This all ties back to the overall goal of ensuring our methods are robust and trustworthy. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. I appreciate your engagement and look forward to working with you further.